Hi everyone. In this demo lecture, we'll test the out of Africa hypothesis. So the plan for today is first we'll see why studying human evolution is important, what efforts have been made in this direction to strengthen our understanding of human evolution. Then we'll see what is mitochondrial DNA, why is it important for the study of evolution. And then we'll see what is out of Africa hypothesis, when was it proposed and why is it important. And then we'll see phylogenetic, a bit about phylogenetic trees, because in this demo we are going to use phylogenetic trees to test this out of Africa hypothesis. And then finally we'll jump into our demo where we'll use some publicly available mitochondrial DNA sequences and build a phylogenetic tree using those sequences. And then we'll see using that tree how we infer that early humans originated in Africa and then they spread to the rest of the globe. Okay, so the first question is why study human evolution at all? So let's start by answering this question. The first reason to study human evolution is to understand our origins and connection with other species. Human evolution provides insights into where we come from and how we have evolved over these millions of years. By studying human evolution, we learn about our relationship with other species, both living and extinct, like Neanderthals and Denisovans. Now, studying evolutionary history helps us answer questions like why certain genes persist, how some diseases evolve, and why certain genetic traits are more common in certain species or in certain population while uncommon in other populations. It helps us understand the development of human intelligence, social behavior and the cognitive traits, and helps us strengthen our understanding that of those cultural traits that actually define humanity. It also helps us explain the genetic diversity that is seen in modern humans, and which we'll also kind of see in our demo today by aligning mitochondrial DNA sequences of humans across the globe from Africa, Asia, Europe and America. Evolutionary studies highlight how humans have adapted to environmental changes over a large period of time, different difficulties like food shortages and etc. Lessons from our evolutionary history can help us tackle challenges like climate change, pandemic, and other environmental stressors. So that concludes that while learning from our past and why studying human evolution is very important. So here I'm going to show you a timeline of when this study of mitochondrial DNA to find our origin and to study human evolution started. So this paper came in 1987, January 1987, Mitochondrial DNA and Human Evolution, which first proposed this out of Africa hypothesis that early humans, which are Homo sapiens, originated in Africa. But this is not the only hypothesis that is present. There is one other hypothesis which is called multi-regional evolution of humans. So this paper came out just 5-6 years after this paper, and then there is genetic and fossil evidence for origin of modern humans and a lot of research which is still going on even in 2000. This paper came in 2002 out of Africa again and again. This paper which came out in 2012, the Great Human Expansion, which also kind of support out of Africa hypothesis. And with all this paper which recently come out probably in 2022, the Human Origin and Migration, deciphered from novel genomic footprints of mitochondrial DNA sequences. So it also supports that humans originated in some parts of Africa. Okay, so we'll see about this timeline. So we see that this is the paper which came out in 2022. We'll also use some publicly available mitochondrial DNA sequences data reported by this paper to build our phylogenetic tree and test the out of Africa hypothesis, right? So before jumping into that, let's complete the rest of the things that we have to do in this lecture before building actual phylogenetic tree and understanding how we can infer out of Africa hypothesis from that. So the first thing is mitochondrial DNA. So it is a small circular form of DNA found in mitochondria, which you all must be knowing by the name powerhouse of the cell. So unlike nuclear DNA, it's inherited exclusively from the mother. So why are we studying mitochondrial DNA only, not the other parts of DNA, the whole nuclear DNA? This is because there are many, so there are some regions. The first is it is inherited only from maternal parent. So there are very less chances of recombination. The recombinations are not that frequent, and there are some occasional mutations only. So the genetic material is inherited from one generation to the next generation with very less changes, right? And okay, next coming on to out of Africa hypothesis. So one thing to point out is just like mitochondrial DNA, in past few years people have also tried to use Y chromosome, because Y chromosome is also inherited from only one parent, that is paternal parent, and that is why undergo very less mutations, very only occasional mutations. Whereas in case of other nuclear, whereas in case of nuclear DNA, other parts of the DNA offspring inherits a DNA which is a mosaic of both the parents' DNA sequences. Okay, now let's jump on to out of Africa hypothesis, what actually this hypothesis is. So it proposes that modern humans, which are Homo sapiens, originated in Africa around 200,000 years ago. This 200,000 is not an exact number. So some researchers claim that it is around 150,000 years ago, some say it's 160,000 years ago, but it is an estimate with a relatively large error. And then it says that a single population of early humans migrated out of Africa and spread across the globe. So, and this hypothesis is challenged by another hypothesis, which is multi-regional hypothesis. But in recent times, with multiple studies coming along, there is much more evidence of out of Africa hypothesis than this multi-regional hypothesis. So what are the evidences that scientists put forward to support out of Africa hypothesis? The first is that the most diverse mitochondrial DNA is found in African population, suggesting that humans originated there, right? And the oldest fossil of anatomically modern humans, the Homo sapiens, have also been found in Africa, which is dating back to over 150,000 years ago. And it's not just mitochondrial DNA. As I said, people have tried using Y chromosome studies, and that have also shown similar results, showing a common African ancestor for modern humans. Okay, and now let's talk about phylogenetic tree, because that is what we are going to use in our demo to see if how do we infer from a phylogenetic tree of mitochondrial DNA sequences that we have a common ancestor originated in Africa. So this on your right you see is a phylogenetic tree. This is a diagram that represents evolutionary relationship between different species or organisms. In this case, it's defining about this is defining a relationship between different humans in across the globe. So almost these are all African genomes, these are all kind of non-African genomes, and you see that as the previous in the previous slide I mentioned that it's the most diverse mitochondrial DNA is present in Africa. So here the diversity is very large, however here the diversity is very less, which are non-African, which kind of supports the out of Africa hypothesis. Now, each branch in this phylogenetic tree represents a common ancestor. For example, if you look at this point, then this is a common ancestor of all of these and all of these humans, right? And the length of branches is also one of the most important factor we need to understand to infer a phylogenetic tree. The length of the branches usually indicates the genetic distance or the evolutionary time. For example, here it's pointed that this mutation event, this event occurred around 3800 and 38,500 plus minus 1500 years ago. Usually the errors in these studies are large. So similarly here it would be around some 60,000 years ago. Yeah, and similarly this event is expected to be around 200,000 years ago. So the higher the longer the length of the branches, the longer the evolutionary time or genetic distance from the root, right? Okay. Now this is a common phylogenetic tree of some species, crocodiles, amphibians, sharks. So yeah, so this is here to show that why phylogenetic trees and how phylogenetic trees are important when we are studying human evolution or other evolutionary studies. So it helps understanding about speciation events. So like some speciation events that occur, for example, it also helps us understand about speciation events, like how different species split, and also the time of those speciation events. Not just speciation events, it also helps us understand about evolution of traits. For example, around what time, after what adaptations, bipedalism occurred in humans or flight occurred in birds. So these are sort of things that can be inferred from a phylogenetic tree. So yeah, that was it about phylogenetic tree. Now it's time to actually construct a phylogenetic tree from publicly available mitochondrial DNA sequences and see what we can infer from that, right? So now let me go to my web browser. So I have already opened this paper, which I also mentioned, come around 2022. There is a minor correction. This was published in 2020, not 2022. Okay. So this paper in supplementary material, you can actually go on this go and read this paper. It's an interesting paper. If you go to supplementary material, okay, yeah, then you will find this Excel file. You can download this Excel file. This has data of publicly available mitochondrial DNA sequences. There are around more than 370 DNA sequences, accession IDs provided in this table. So if you look at this table, this is like the region from where the person was selected for sequencing their mitochondrial DNA. For example, here it's Middle East, South Asia, Europe, in Europe, Georgia, Europe, France, and so on. And these are accession IDs. So how do we get the actual mitochondrial DNA sequences from this accession ID? For example, I can just copy this accession ID, go to my web browser and search for NCBI website, because this data is publicly hosted on NCBI website. Okay, now let me go to NCBI website. This data is publicly hosted on NCBI website. Since this is DNA sequences, I'll go to nucleotide. Okay, I'll copy paste that ID and search for that particular ID. Okay, now here is the information about that particular ID, mitochondrial DNA sequence. I can click on FASTA and I'll get the FASTA sequence. Yeah, this is the FASTA sequence. One way is you can copy this, or other ways you can click here, click on file, like you want to download it as a file, or copy on clipboard, that is also fine. Here you can select the format. I only want the FASTA file. If I click on create file, it will download the file. Yeah, it has downloaded the file in my system. But if you see, there are around 370 genomes. Downloading each one by one would take a lot of time. So there is one tool called Entrance Direct. You can download this tool from using Conda. This package is available on BioConda. Install it, and then there is a very simple command you can use. I have already downloaded all of these sequences. Then let me just show you how these sequences would look like. So if I open my script, let me show you by opening my script. Okay, so this is a simple script like efetch is the command, then database is nucleotide, which we also saw on NCBI website. Format is FASTA. ID is, you can provide ID here. I have listed all those IDs, and then you can append this on a single file, which is sequence.fasta. I'll show you how the sequence.fasta file would look like. Let me click on sequence.fasta file. Okay, so this will be the ID, and the sequence.fasta file is shown similarly as it was shown on NCBI website. So there will be all the sequences. But one thing that I have done here is, so these are ID names, and we are not very good with remembering the ID names by remembering the mapping. Yeah, we are not good in remembering this mapping from ID to where the person, what was the geographical location of person who was sequenced with this ID. So what I've done is, I have replaced these names. For example, ID name replaced by the geographical location. Here it's Africa. In some cases it will be, yeah, it's a different place in Africa. In some cases it will be Europe, Asia, and so on. So I have replaced all these ID names from these, so that when we build our phylogenetic tree after constructing the multiple sequence alignment, we'll only see those names which are here, which are their geographical locations, not the IDs. That is one thing. Now let's directly jump on to do a demo. For that, let's go to, okay. In our previous demo also we did multiple sequence alignment, and for multiple sequence alignment we used a tool called Clustal. But now since we have not 4, 5 or 6 sequences, we have 370 sequences, and also the sequence sizes are not 10 or 20, they are large. So we are going to use a different tool which is called MAFT, MAFFT. So again, there are many options here. You can copy paste a protein or DNA sequence here, or you can choose a file, a FASTA file or plain text file, anything would do. And there are some other options about the input. When you get the output Clustal format, what is the title length you want? Is it 10, 100, 20? You can also specify the job name here. By specifying job name, you will be able to recognize if you are submitting multiple jobs. You can also provide your email address here. For example, if your job is very large, this tool is made for large sequences, and compared to what this tool is made for, our sequences are not that large, so will not be requiring this feature. It will be just 10 or 20 seconds. Now there are multiple other options as well about strategies. In our previous demo also we talked about multiple sequence alignment strategies, progressive and iterative methods. You can select any of these methods. Then there are these parameters of scoring matrices. For amino acids is BLOSSOM, for scoring matrice in nucleotides is PAM. Similarly, there are multiple options. You can play around these options and see what fits the best for you. For me, I'm going to use the default option. Now let me upload and click on submit. Right. While it's running, let me show you one more thing. There are 370 sequences which are corresponding to all Homo sapiens across the globe, and there are these 6 outgroup as well, which are baboon, chimpanzee, and gorilla. There is something called bonobo. So I have built 2 FASTA files in which there are only one in which there are only 370 sequences, one which includes all of these 6 sequences as well. So we'll build a phylogenetic tree using both of these FASTA files. In one, we expect to see just the alignment of all of these. In one, we expect to see that how our relationships are with these outgroups, which are chimpanzee, gorilla, baboon, and bonobo, and so on. So let's go ahead and do that. Okay. So it has created this multiple sequence alignment. This is the multiple sequence alignment format. These are all the ID names, right? So there are many. You can download it in cluster format, in FASTA format. A zip file will be downloaded. But we have to create a phylogenetic tree as well. So let's go ahead and click on phylogenetic tree. Okay, to create phylogenetic tree as well, there are different methods. You can choose these features. You can play around these features. I'm going to use the default features only. So let's click on go. So it will just take a few seconds to create the phylogenetic tree. Okay, it has created a phylogenetic tree, and we can view this phylogenetic tree in view.io. Okay, so there are two versions. One is a resolved tree, which will be like with complete all the branches. Let's since there are 370 sequences, let's see a collapsed tree, which is fast as well. Okay, so if you look at this, this is so like this baboon is very far from all other species, Homo sapiens, chimpanzee, and gorilla as well. And then the second is gorilla. So if you look at this point, so at this point, this was some common ancestor of all the gorillas, bonobo, chimpanzee, and Homo sapiens, right? And similarly, then if you look at the genetic diversity, as I mentioned, this length defines the time of evolution, right? So then this event, so if you look at the time of this particular event or time of this particular event has occurred like maybe millions of years before this particular event. So here we are not able to see clearly about all the Homo sapiens, their differentiation as we saw in our PowerPoint presentation. So what I'm going to do is again go to MAF, another tab. This time I'm going to select the sequence which only contain the mitochondrial DNA sequences of Homo sapiens, not these outgroups, right? And with that we expect to see in more detail about what we can infer, that how early humans, the Homo sapiens, originated in Africa, then spread across the world. We'll see how this. So this usually takes around 20 or 25 seconds. Let's see. Okay. Yeah, as I mentioned in background, it does first the multiple sequence alignment and a phylogenetic tree using different algorithms like maximum parsimony, maximum likelihood method, and so on, is built after this multiple sequence alignment. So we got the multiple sequence alignment. Then let's click on this phylogenetic tree with just the default options. Let's build our phylogenetic tree. Okay, so we have our phylogenetic tree. Now let's see again this in view.io. Okay, now it's we can clearly see, because all of these are Homo sapiens. If you look at this graph, then all of these are African genomes. All of these are also African genomes. Here itself you can see there is a lot of genetic diversity across African genomes, but that's not it. If you go here, these are also, all of them are African genomes. All of these are African genomes. These are African genomes, and even here these are some African genomes only. And these are some, so like here you can see these are some Europe, here it's Middle East. But if you look at look overall, if you look at this phylogenetic tree, then you would realize that there is a lot of genetic diversity across African genomes, and yeah, and also there is a long series of time starting from here to here where African genomes existed. However, if you look at these, so looking at this phylogenetic tree, if you zoom into any of these, so these are collapsed, for example, let me try to zoom into this particular Let me click on expand. Okay. Okay, let me try to click on expand all. So again, it has expanded all of them, so it looks a little cumbersome, but let me collapse some of these. Okay, and now things are a bit clear. Let me collapse some of these as well. Okay, now things are a bit clear. Here also you see one of the African genome, Europe, Africa, Europe, Middle East. And similarly, if you try to zoom into, for example, this particular area, then you will see Europe, Finland. Similarly, if you zoom into, because there are 370 sequences, so there is a very large number of sequences. So for example, if I again zoom into this particular area as well, here also you will see Asia, America, North America, Asia, Chinese, and so on. So this becomes a little cumbersome, but if you again zoom in instead of zooming out, then it's much of a cleaner tree. But we have zoomed out and see that all of these are from all across the globe, not just Africa, and also see some African genomes here. So this is very clear from this phylogenetic tree that there is a whole lot more genetic diversity when it comes to African genomes than the genomes that are all across the globe except Africa. So yeah, so this supports the hypothesis that at some point in time, yeah, if we look at our demo, that at some point in time, which is around some 200,000 years ago, early humans existed here, and then at some around 100,000 years or some 60,000 years ago, people started migrating out of Africa and then populated these regions all the way to all these regions. So using this phylogenetic tree, we have concluded that the phylogenetic tree of mitochondrial DNA sequences is supporting the out of Africa hypothesis, and using this phylogenetic tree, we have concluded that our closest common ancestor among these groups are chimpanzee, then it's bonobo, then it's gorilla, and then very far it's baboon, right? So yeah, that was it for this demo. I hope you all enjoyed this demo, tracing your roots. Yeah, so thank you everyone.